my screen with the title slide?
Yes.
Sharing. Okay, good. All right, so let me first, I first have to hit the record button. Well, let's see. Record.
Recording in progress.
Record to the cloud. Okay. That means that we will be able to send you a link probably later today, but we'll see in terms of when it will be available. So we're going to first start, we're going to do it in the same order we did it in class. The first part of the talk is going to be about the GI tract, the tube that runs from the oral cavity down through the anal canal. And when you're approaching histology, you'll notice particularly in the GI tract that there's a general pattern of organization. So we're going to go over that first. And as you go down the tract, just keep in mind that you want to pay attention to where that pattern is broken. So in much of the tract, it will be the same pattern of organization, but there will be places where various different parts of that pattern are altered. And it's always important to pay attention to those things where the pattern changes. So let's first look at this slide, which is a slide demonstrating the general structure of the gastrointestinal tract. So you can see that this is a cylinder that we have opened by cutting out a portion of the wall of the cylinder. And the lumen is in the center. Let's see if I can get a laser pointer. Lumen is in the center here. And as we progress out toward the outer edges of the cylinder, we go through multiple layers. Remember, histology is often a question of learning the layers in order. So the layers going from inside to outside are mucosa, submucosa, muscularis, which is also termed sometimes muscularis externa or muscularis propria. Those are synonyms. And then finally, on the outer layer, the serosa or adventitia, which would be here. Now, this little drawing is an example of a segment of the GI tract which has a serosa, because you can see that the outer layer is shiny because it has a layer of mesothelium on the very outside. And it has that because this portion of the organ is wrapped in a fold of the peritoneal membrane. And the area of the peritoneum that suspends that segment of the GI tract is called either a mesentery, if we're talking about the small intestine, or mesocolon, if we're talking about the large intestine. Interestingly, the little vermiform appendix, which is an outpouching from the cecum at the beginning of the large intestine, also has its own little suspensory ligament of peritoneum, which is called the mesoappendix. Okay. Well, if we break down each layer, we can subdivide them. So a lot of times people get confused about this. The mucosa is defined as epithelium, lamina propria, and muscularis mucosa. Okay, so the mucosa in this drawing is this tan bordered zone here, which has a little red line at the base. So, and actually, if you look very carefully, it has sort of a browner edge to it. So the brown edge at the very luminal edge is the epithelium. The lighter tan is the lamina propria, and the little red line at the base there is the muscularis mucosa. Now, deep to that in this drawing is a purple, light purple or violet layer, and that is meant to represent the loose areolar connective tissue with blood vessels, lymphoid cells, nerves of the lamina propria. I'm sorry, of the submucosa. Even I get confused about which layer is which. So the second layer is submucosa, and that is illustrated here as this lavender layer. Now beneath that is this double-coated layer of muscle. That is our muscularis layer, and typically in the GI tract it is 2 layers. There will be exceptions, so you have to pay attention to the exceptions. But the inner layer is circumferential. You can see the fibers running around the circumference of the tube here, and the outer layer is longitudinal. So in this layer, the drawing is showing us cutting these muscle fibers in cross section. So a two-layered muscle coat, and outside of that, if the section of the GI tract is intraperitoneal and covered by a fold of the peritoneal membrane, it has a serosa. And if it is extraperitoneal, not covered by a membrane, for instance, an example of that would be the esophagus, which traverses the soft tissue in the center of the thorax, then we call that outer layer the adventitia. Okay, a couple other structures are mentioned here that are important. In the submucosa, we sometimes have submucosal glands. These are limited in distribution. We have them in the esophagus. We have them famously in the duodenum. But most of the other areas of the GI tract lack submucosal glands. We also have a submucosal nerve plexus, which is eponymously termed the Meissner plexus. That is the name, the Meissner plexus. And that is in the submucosa. And that extends all the way pretty much throughout the GI tract. So if we ask you about a nerve plexus in the submucosa of the ilium, it would be Meissner. If we ask you about a submucosal nerve plexus in the duodenum, it would be Meissner. Okay? The muscularis propria has its own neural plexus, and that is between the two major layers of the muscularis, the inner and outer layers. And this nervous plexus is called the Auerbach's plexus. That is misspelled. The H is floating down at the bottom line there. I don't know what happened there, but it's Auerbach's plexus. Okay? So that is the general plan of the GI tract. So as we go through from oropharynx down to the anal canal, pay attention to where it changes, okay? So we'll start the oral cavity, okay? Oral cavity, we include the lips, the tongue, the oral mucosa, and the minor salivary glands. In this class, we don't talk about the teeth. There will be no questions on this exam about teeth. Okay, so you don't have to review that or read about it. Okay, let's see. Let's talk about the lips. So this is the section from, I think, the lab session. In the center, we have a sagittal section through the free edge of the lip. And if you remember, there are several variations in the mucosa of the lip. The outer part of the lip is skin. The inner part is oral mucosa. Now, the oral mucosa is predominantly non-keratinizing stratified squamous, predominantly. There are exceptions individual to individual, and there are some areas that tend to be keratinized. These are referred to in Juncaria as the masticatory mucosa, and mostly refers to the gingiva and the hard palate. But there are variations in different individuals. For our purposes, we're just going to say that the predominant mucosa is non-keratinized stratified squamous. On the skin side, you always recognize skin because you can identify hair follicles in most areas of thin skin. We learned that a couple of weeks ago. On the oral mucosa side of the lip, we have a minor salivary gland labeled G in this image. So that is a feature that we see throughout the oral cavity, is unencapsulated minor salivary glands. We also have a transition zone of the epithelium called the vermilion zone, labeled V, and this is where you put chapstick on your lips or lipstick on your lips. And this is a particular section of the epithelium where the epithelium is thinner. There is a well-developed vascular component right underneath the epithelium, and that's what makes it a more reddish color. It also is has extensive sensory innervation, so we see modifications like Meissner corpuscles immediately under the thin epidermis of the vermilion zone of the lip. Okay? Let's look at the tongue. We spoke about the tongue, and we know that the tongue has two big functional zones. The anterior two-thirds of the tongue we refer to as the oral tongue or the mobile tongue, and this is where we find most of the lingual papillae. The sulcus terminalis is this V-shaped groove at the two-thirds to one-third boundary zone and separates the anterior two-thirds or oral tongue from the posterior two-thirds, which we also call the base or the root of the tongue. And this is where we have one of our tonsils, our lymphoepithelial organs. This is the location of the lingual tonsil. Now, we spent a lot of time in class talking about the different types of taste buds. Just remember there are 4 taste buds: filiform, fungiform, circumvallate, foliate. 3 out of the 4 have taste buds in them. The one that doesn't is the filiform, and this is the keratinized papilla of the tongue, lacks taste buds, and it gives the surface of the tongue, the dorsum of the tongue, its rough texture. Okay. Now, the specialized sensory organ that's involved with the taste buds is shown on the diagram on the right side of the slide. This is the taste bud. And we know the taste bud is sort of analogous to the specialized epithelium we studied in the roof of the nasal cavity, the olfactory epithelium, because the taste bud is a collection of cells that includes sensory cells, sometimes called gustatory cells because they're involved in taste, a complement of supporting cells, which some sources refer to as a sustentacular cell, basal cells at the base of the taste bud, and modifications at the apical end of the gustatory cells, including microvilli, which extend out through a pore in the overlying squamous epithelium. And this is where the molecules that we taste actually interact with those sensory cells, is at the microvilli poking through that taste pore. Okay, and of course, once these gustatory cells depolarize, they send that impulse through sensory nerves at the base of the taste bud. This drawing is meant to show the pink here is epithelium. So, you know, the taste bud is contained within the thickness of the epithelium. Okay, that's pretty much what we're focusing on in the oral cavity. Let's move on to the esophagus. Okay, this is really the first part of our tube-like stretch of the GI tract. And once again, we have 4 layers. So we have the same basic plan here: mucosa, innermost layer, and then moving toward the outside, submucosa, muscularis, muscularis externa, and in the esophagus, adventitia, because the esophagus is extraperitoneal. It traverses the thoracic cavity through soft tissue in the mediastinum, the central portion of the thoracic cavity. So let's just break this down a little bit. We know that the mucosa has 3 components from inside to outside: epithelium, lamina propria, muscularis mucosa. Now, one slight variation in the esophagus is the muscularis mucosa is incomplete. So if you take a sample of the esophageal mucosa, at one place you may see a defined muscularis mucosa, but in another sample you may not, because that structure is incomplete in the esophagus. The submucosa is interesting because this is one of the sites that sometimes has mucus glands in the submucosa. If they're in most places in the esophagus, we just refer to them as esophageal glands. And we have a special name for them if they're close to the esophagogastric junction. Then we call them esophageal cardiac glands. But all these submucosal glands are similar. They're all mucus. And of course, the mucus predominantly provides a lubricating function for the esophagus. The muscularis externa in the esophagus also is a break-in pattern. It follows the pattern in that it has an inner circular layer and an outer longitudinal layer. But where it differs from the rest of the tract is that the muscle coat is a mixture of skeletal muscle and smooth muscle. The upper third of the esophagus is almost entirely skeletal muscle in the muscularis. The middle third is a mixture of skeletal and smooth muscle, and the lower third is almost entirely smooth muscle. So if you look at the image on the bottom of the slide there, this is from Junqueira, and it shows a little bite of the esophageal muscularis from the middle third. And we know it's the middle third because we can identify skeletal muscle cells and smooth muscle cells. To identify skeletal muscle cells, if you're given a histologic section, look for this kind of appearance. This is really a cross-section of a skeletal muscle fiber. You see, it has this pink sarcoplasm, and the nucleus is invariably at the periphery of the cell. You see that pattern? So if you see that pattern of these very pink-looking cells with peripheral nuclei, you have skeletal muscle. On the other hand, the upper left corner of this image shows spindly cells in muscle tissue. This is smooth muscle. So this mixture of skeletal and smooth muscle tells us this sample is obtained from the middle third of the esophageal muscularis. Okay? One wrinkle about the adventitia in the serosa is that at the very distal end of the esophagus, it pokes through the diaphragm at the crura, and then it acquires a little bit of a surface of peritoneal covering. So the very most distal part of the esophagus, like 1 or 2 centimeters, is actually intraperitoneal and therefore has a serosa. Okay? Let's move on to the stomach. The stomach is a break in our tubular pattern because the stomach is not shaped like a cylinder. The stomach is shaped like a sack, or like a leather wine bottle that somebody might carry their wine in up in the Alps. Have you seen these things? Right? It has a narrow end and a bulbous end at the other extreme. And this shape is important in terms of the function of the stomach because unlike some other parts of the GI tract where the major function of the muscle coat is to move the contents of the GI tract along the lumen, the stomach also has a role in breaking up the food that we eat. So the stomach has to be able to not just move food along, but also shake it and physically disrupt it. So as a result, the muscularis externa of the stomach has a different structure. Instead of the typical 2-layered structure, the stomach has a 3-layered muscle coat. The inner layer is obliquely oriented fibers. The middle layer is circular oriented fibers. And the outer layer is longitudinal oriented fibers. And this 3-layered structure facilitates the stomach shaking the contents and breaking it up so that the food is more available to be acted upon by the digestive enzymes that are produced by the mucosa, both in the stomach and distally in the small intestine and in the pancreas. Okay? The stomach, unlike most of the esophagus, is intraperitoneal, so it is covered by a serosa, right? The mucosa of the stomach is probably more complex than the mucosa in any of the other segments of the GI tract. So we're going to get into that next, about the different patterns of mucosa lining the stomach. But just like the other sites in the GI tract, the mucosa is epithelium, lamina propria, although in the stomach it's a very small amount of lamina propria, and muscularis mucosa. So let's take a look at why the stomach has different patterns of mucosa. So the stomach actually has different geographic regions. This drawing shows the general shape with a lesser curvature on one side and what we refer to as a greater curvature on the other side. This is the distal end of the stomach, right? Can you see my cursor there? Right? Distal end of the stomach feeds into the duodenum, and the proximal end of the stomach receives contents from the esophagus. Within the stomach, the anatomic regions are cardia, which is a very small area, about 1 or 2 or 3 centimeters wide, right at the esophageal gastric junction. The second area is the fundus, which is a somewhat larger area, but represents that portion of the stomach which exists above or cephalad toward the head from the GE junction. Here's the GE junction. Everything above that is the fundus. Everything below that, or most below that, until we get to this region here, the antrum. This region from the cardia down through the notch at the incisura is the gastric body or corpus. This is by far the largest geographic zone of the stomach, and it's also the most complex mucosa. You see that the fundus and body both are lined by an oxalic acid-producing mucosa. Let's see, we have Dr. Malvar is raising her hand. Dr. Malvar, are you raising?
Hi, Dr. Bannon. Could you use a red pointer?
I can. I'm very awkward, but there it is.
It's okay. Yeah, I think it's the black tiny pointer just is not as visible. Thank you.
Okay, there you go. There's my red dot. Okay, so just going over these geographic zones again in case it wasn't clear. Cardia, right near the esophagogastric junction. Fundus, the portion of the stomach that is cephalad or above the cardia. Body, the area of the stomach between the cardia and the notch on the lesser curvature, which is the largest area of the stomach. The antrum, which is a funnel-shaped area at the distal end, which narrows the diameter of the stomach down from the largest diameter at the body down to the narrowest portion of the pyloric channel. Sometimes this area here is called the antropylorus together. Okay. Now, in terms of the mucosal coverings, there are basically two types of mucosa. This oxalic or acid-producing mucosa is the most common type, and it's the most complex type. This covers the fundus and the body of the stomach. Then there's basically a second type of mucosa, which is sometimes referred to as antropyloric, but is very similar to the mucosa in the very small cardiac region. And this is a mucosa that is rich in mucus glands. And we're going to talk in a little more detail about how these two mucosas, what their features are and how they differ. Okay, so let's take a look at that. Let's first look at what we call the oxalic or acid-producing mucosa that covers the fundus and the body. So these are H&E stained sections of this mucosa. And the image on the left here is broken down into the different geographic regions of these gastric glands. So I think there's often some confusion about this because, well, I'll show you why in a second. This is a very thick mucosa, and there are basically three different zones. There are the surface and gastric pits. So the surface is the very top. Gastric pit is when the epithelium dips down into the lamina propria. So that's the very most superficial part of the mucosa. The next region down is referred to as the neck or the isthmus region of the mucosa, right? And then the deepest region is, I think confusingly, referred to as the gastric glands. This whole thing is really a gland, okay? But then when we break down these glands that line the stomach into different zones, we call the deepest zone the glands, okay? Now, the cell types are distributed geographically in the different zones. So in the surface and gastric pits, the predominant cell type is the surface mucus cell. In the neck region, the predominant cell type is the mucus neck cell, but not shown on this slide, but very important, is that this is also the location of the stem cell. The stem cells in the neck or isthmus of these glands can replace any type of epithelial cell if it's damaged or lost. In the gastric gland region, we have predominantly a mixture of parietal cells and chief cells. The parietal cells in the H&E stain section stain pink. They usually stain brightly eosinophilic. You can actually pick them out in this image, like this one here is a nice one. So the cytoplasm is bright pink. Pink stands for parietal, okay? And the bluish cell, which is the other cell type that's major in the gastric glands, is the chief cell, okay? There's another cell that's in this zone that we don't have here, which is the enteroendocrine cell that we're going to speak about. And at the very base of this image are a few strands of smooth muscle that are labeled here as MM, muscularis mucosa. So this is showing you the entire thickness of this very thick and complicated mucosa that lines the body and the fundus, and that we refer to as oxyntic mucosa. So let's break down those cell types, okay? So this is a very important slide because understanding the way the GI tract functions, really, it's very important to understand the different cell types that are in the different glands at different levels of the GI tract and know what they do, any products they produce, and what their function is. So let's go over these cell types from the top to the bottom. At the top, surface mucus cells. These cells secrete mucus. There's a very viscous mucus that sticks to the surface, and it forms a protective alkaline mucus layer. So this is a layer that protects the surface of the stomach mucosa from the acid that the mucosa itself will produce. The next level down, the isthmus or neck, has mucus neck cells, which also secrete mucus, although it's a less thick mucus, and it's a little more acidic than the mucus that's produced on the surface. And of course, as we also said, the very important stem cells are also localized in this area, and they are capable of replacing any of the epithelial cell types in these gastric glands. Now down to the gastric gland region, down here at the base, there are three major cell types here. Parietal or oxyntic cells, those are the pink ones, acidophilic on H&E stain. They secrete hydrochloric acid and, very importantly, intrinsic factor. I think that in biochemistry and maybe in physiology, you folks have recently talked about pernicious anemia. And pernicious anemia very much involves the absence of intrinsic factor. Intrinsic factor is produced in the stomach, but is required for B12 absorption in the small intestine. Okay? And of course, hydrochloric acid is very important in terms of digestion, and that is a product of the parietal cells. Now, the other component, one of the other major components that the gastric mucosa contributes to digestion is digestive enzymes. And these are produced in the chief cells, which stain basophilic or blue on the H&E stain section. And they make pepsinogen and a precursor lipase. You'll notice this is a common pattern in the GI tract where the cells that are producing digestive enzymes synthesize proenzymes that are then activated when the proenzyme reaches the lumen of the organ. So the same is true here in the stomach. The chief cell synthesizes pepsinogen and a lipase precursor that get activated when they hit the lumen of the stomach. The third category in this particularly complicated gland are the enteroendocrine cells. Now, these are sometimes referred to as enteroendocrine cells is the most general term, but you'll also see them referred to as enterochromaffin cells or argentophin cells. And as you've already learned when we studied histochemistry, we often name stains by what the chemistry of the stain is. So we call it an enterochromaffin cell because they're cells associated with the intestine that have an affinity for chromium salts. So enterochromaffin is the name that we give those cells. Actually, that's misspelled on the slide. It's chromaffin, not chromatin. And then argentophin cells are cells that have an affinity for silver cells. We can also identify these cells by staining them with immunohistochemical techniques for their product. Now, we're focusing on two examples in the stomach. EC cells, the EC stands for enterochromaffin, and these cells secrete serotonin, which has a role in increasing the motility of the gut. And the other one we're focusing on are the G cells, and these are mostly in the antropylorus region of the stomach, and they secrete gastrin. So if you wanted to identify all of the G cells in the stomach, you could use an immunohistochemical stain directed against gastrin and identify G cells that way. Let's take a look at one of these really important cells in the gastric gland, the parietal cell. So this is a drawing from Junqueira that contrasts the parietal cell at rest and the parietal cell active. Parietal cell gets activated when we have a meal. So when there's food in the stomach, the parietal cell is stimulated to produce acid. And the upper portion of this image, this half here, shows the parietal cell when it is activated. It develops a very prominent intracellular canaliculus that is lined by MV, microvilli. And because this process involves pumping ions to create hydrochloric acid, that pumping ions against gradients requires energy. So there are also numerous mitochondria in these parietal cells. Okay, so those are the most important ultrastructural features of the parietal cell. An intracellular canaliculus with microvilli and mitochondria. Now, the parietal cell also synthesizes protein, so we do have the anticipated protein synthesizing machinery we often have, such as rough endoplasmic reticulum and Golgi apparatus. But the thing that's distinctive about the parietal cell is this ability to produce large quantities of acid through the intracellular canaliculus with its microvilli and numerous mitochondria. If we look at the ultrastructure of one of these gastric glands, so this is a cross-section through a deeper segment of this gland, the zone that we call the gastric gland. So we have a mixture of chief cells, there's a nice one is right here, and parietal cells. There's a nice one here, and maybe, I'm not sure if this is one cell or two here. It's maybe one large cell. But the feature you see in the parietal cell that makes it unique is this space. This is an intracellular canaliculus, and you see that it has villi lining the surface or the edge of the canaliculus. The same is true with this one over here, right? Now, in contrast, the chief cell here, right, has a zone at the basal part of the cell that has lots of rough endoplasmic reticulum. I think you can see the texture here is sort of lamellar because you're seeing packed stacks of rough endoplasmic reticulum. But you also have secretory granules on the apical side of the cell. This is another chief cell over here, which shows lots of secretory cells on the apical half and some rough endoplasmic reticulum, which is hard to visualize here, but it is also there. Now, the third cell type is also visible in this section, and that is here. That is an enteroendocrine cell. And this cell here does not seem to communicate with the lumen. So this is what we refer to as a closed cell type of enteroendocrine cell. And it has the hallmark of the enteroendocrine cells, which is the presence of neurosecretory granules. See these dark electron-dense granules on the basal part of the cell, right? And remember, function often dictates form, right? And if you compare, remember the chief cell is basically secreting its product into a lumen on the apical side of the cell. So its secretory granules are on the apical half of the cell. But in contrast, the enteroendocrine cell is an endocrine cell. So it produces products that it secretes through the basal membrane into the bloodstream. So its granules are on the basal half of the cell. Okay? Now, I told you that there are different types of mucosa in the stomach, and we've been focusing on the most complicated one that covers most of the stomach, the actinic mucosa that covers the fundus and the body. Now, the other type of mucosa is illustrated here, and this is an example of the mucosa that covers the antrum and the pylorus, sometimes referred to as the antropyloric region of the stomach, but a very similar mucosa covers the cardia, the very narrow zone just beneath the gastroesophageal junction. And this mucosa differs from the one we've been just looking at because its glands are much simpler. Instead of these complicated glands that produce acid and digestive enzymes and mucus, these glands just basically produce mucus. They're pyloric mucus glands. They also produce lysozyme, which has an antibacterial effect. And they also contain in the antropylorus enteroendocrine cells, which are called G cells that secrete gastrin. Okay. Now, the major distinction between the mucosa of the antropyloric region and the mucosa of the cardiac region is that cardiac region mucosa does not have G cells. Okay. Otherwise, it's a very similar mucus gland mucosa. Right? You'll notice that in neither of these regions do we have parietal cells or chief cells, the characteristic cells present in the oxyntic mucosa. Okay? All right, we're ready to move down to the small intestine. Any questions about the stomach before we move on? No hands raised? Pretty straightforward. Okay. Small intestine. Okay, so we got a nice cartoon on this slide, all the way over here, that shows the position of the small intestine in the peritoneal cavity. And if we take a small sample of this and blow it up into a larger drawing, we see what the wall of the small intestine looks like. Okay, so in this picture, we have mucosa, we have lamina propria. So this is just the mucosa here, and this is muscularis mucosa at the base. Okay, now this is also showing one of the characteristic features of the small intestine, which is that the mucosa is thrown up into structures we call villi. These are finger-like structures with a coat of epithelium on the outside and a core of lamina propria in the middle of the finger. Okay, and of course at the base of the mucosa is the muscularis mucosa. We call this structure, the finger-like structure, the villi, but where the epithelium turns down into the lamina propria, we call that segment the crypt. So the small intestinal mucosa is composed of villi and underlying crypts. The crypts have an eponym, Lieberkühn, the crypts of Lieberkühn. Okay? Now let's talk about the constituent cell types in the small intestinal mucosa. So we're going to start from the surface. The surface mucosa is columnar absorptive cells, also called the enterocyte, the epithelium cell that is the predominant epithelial cell in the small intestine. This cell has luminal microvilli that are apical modifications that serve to greatly increase the surface area, which facilitates the major function of the small intestine, terminal digestion and absorption of nutrients. Okay. Now I want to call your attention to something, which is that at first look, these columnar cells resemble the columnar cells on the surface of the stomach and the columnar cells on the surface of the colon. But if you look at their names, they are subtly different. These cells are referred to as absorptive cells, which reflects both their surface modification and their function. Their major function is facilitating the last steps of digestion and, very importantly, absorbing nutrients. In contrast, the cells lining the surface of the stomach and of the colon, the stomach cells are called surface mucus cells. They're not called absorptive cells because their main function is producing lubricating and protective mucin. Okay, the same thing is true in the colon, where they are mucus cells at the surface, and they're absorptive cells also, but they use a different mechanism. We'll get to that in a minute. In the colon, the mucus cells are goblet cells. Now, in the small intestine, the second cell type is the goblet cell. And this is responsible for secretion of mucus, which also forms a protective function in the small intestine. The third cell type is the enteroendocrine cell, and we have spoken about two of these. There are numerous examples of these, and there is a chart in Junqueira with numerous examples, but the two we focused on in the sessions are the eye cells, which secrete cholecystokinin, which is important for stimulating pancreatic digestion, enzyme synthesis and release, and importantly, bile release from the gallbladder. And the second cell type is the S cell. And these cells secrete secretin, which stimulate the pancreatic ducts to produce bicarb and water. Now, the next cell type is a stem cell, and here it's listed as the regenerative cell, and we can call it the stem cell. But these cells are located, unlike the stomach, they're located in the base of the crypt, okay? And like the stem cells in the stomach, they can produce any of the epithelial cells in the small intestinal mucosa. And the final cell type is the Paneth cell. And this is also illustrated in this inset on the slide with this very high power blow up of a hematoxin eosin state section. Paneth cells are easy to identify because they've got these really prominent red granules in the apical part of the cytoplasm. These are cells involved in innate immunity, and these granules contain and release antibacterial products, including lysozyme, defensins, and phospholipase A2. Okay, so those three things are the major products that we find in the granules in the pant cells: lysozyme, defensins, and phospholipase A2. Okay, so when we get a sample from the small intestine, there are three segments of the small intestine: the duodenum, which is from proximal to distal, duodenum, jejunum, and ileum. Each of the three segments have distinctive features histologically. So let's just go over some of those quickly. On the left image, we have the duodenum, and the duodenum is characterized by very prominent submucosal glands. Remember at the beginning, I said there are only a few sites in the GI tract that have submucosal glands. We already mentioned the esophagus. This is another one, the duodenum. And in this image, the muscularis mucosa, which is the deep boundary of the mucosa, is right here. It's very subtle, but it's these spindly cells in this very thin layer which separates the mucosa above it from the submucosa below it. But in this tissue, the submucosa is packed in this section with glands. And these are the Brunner's glands, whose function is to produce an alkaline secretion that helps to neutralize the gastric chyme, which is flowing into the small intestine from the stomach. The middle segment of the small intestine is the jejunum. And the jejunum, in a sense, is characterized histologically by a negative feature. It lacks both submucosal glands, and it lacks the characteristic feature we see more distally, which is the lymphoid tissue that we see very prominently in the ileum. The jejunum also has very distinct and well-developed circular folds, which are called plica circularis. So that's another histologic feature of the jejunum. And the most distal segment is the ileum. And the ileum's characteristic histologic feature is the presence of these lymphoid nodules, which we refer to as Peyer's patches. This is the very distinctive feature of the ileum. You can see the ileum still has intestinal villi, like the remaining parts of the small intestine, but it does have this very prominent lymphoid tissue that we call Peyer's patch. The lymphoid tissue has a very distinctive cell contained in it, and that is the M or microfold cell. Let's just talk a little bit about modifications of the small intestine in order to increase its surface area. Now, we already mentioned that the prime function of the small intestine is terminal digestion and absorption of nutrients. And these are functions that are dependent on interaction with the mucosal surface. So the small intestine is modified at three different levels of observation in order to maximize its mucosal surface. So let's look at those levels. The first level is the gross level. That's shown at the left side, and this is where plica circularis is a modification that increases our mucosal surface area. Plica circularis refers to the transverse circumferential folds of the mucosa and the submucosa lining the small intestine. So if we cut a section through one of these plica, see, this is the way the entire small intestine looks. You can see these circumferential folds on the mucosal surface. If we cut across those circumferential folds, it will look like this. And so the key with identifying a plica on a histologic section is that it is a finger-like structure in which the outer surface is lined by mucosa, full thickness mucosa, and the core of the finger is submucosa. Okay? Now, the next level of modification is visible at the level of light microscopy. So we have to magnify the mucosa quite a bit to see the next level of modification. And this modification is the intestinal villi. So these are also finger-like forms, right? But in this case, the finger is lined by epithelium, and the center of the finger is lamina propria. So the entire finger is a portion of mucosa, right? And at the base of the finger is the muscularis mucosa. Okay? The third level of modification is the ultrastructural level. So if we look carefully at these cells at the very luminal edge and we magnify tens of thousands of times, we can see that the apical membrane is modified to have these microvilli, these protrusions of cytoplasm with actin filaments that increase the amount of the surface membrane dramatically. So these are three levels of modification, all of which have the role of increasing our membrane surface area. Gross level with plica circularis, light microscopic level with intestinal villi, and ultrastructural level with microvilli. Okay? MALT, mucosa-associated lymphoid tissue. When we're talking about the different segments of the small intestine, we mentioned that the ileum, the most distal segment, is characterized by its very prominent lymphoid tissue. So this tissue is a mucosal-associated lymphoid tissue, or MALT, in the Peyer's patch. And this is a histologic section that just shows you the mucosa of the ileum. And you can see this very prominent lymphoid tissue, and it's actually typically both in the mucosa and in the submucosa. It is characterized by this very unique cell type, which is the microfold cell, which is characterized by having an intraepithelial pocket in which there's a break in the basement membrane that allows immune cells, both lymphocytes and antigen-presenting cells, the dendritic cells, to enter into this pocket where they are very physically close to the lumen of the intestine, so that antigens can get through transcytosis, can move through this very thin portion of the cell and interact with the immune cells. And once they do that, they stimulate the immune system both in the Peyer's patch and in the regional lymph nodes. Okay? Now, in the same geographic area, we find the vermiform appendix, right? Vermiform appendix is a blind evagination from the cecum. So it's actually attached to the beginning of the large intestine, not the small intestine. But this also contains a significant portion of lymphoid tissue. So these two areas, which are physically close to each other, contain a significant component of MALT for the gastrointestinal tract, the terminal ileum and the vermiform appendix. Okay? If we look at the structure of the appendix, it's like a mini segment of the intestine. It has a serosa on the very outside, which is here. It has a two-layered muscularis or muscularis externa with an outer longitudinal layer and an inner circumferential layer. It has a submucosa where some of our MALT is located. And it has an mucosa, which also contains lymphoid tissue as well. You'll notice that the mucosa actually has a flat surface, so it does not have intestinal villi like we see in the small intestine. Okay, it does not have intestinal villi. All right. Moving on, the large intestine. Now, we talked about the main function of the small intestine being terminal digestion and nutrient absorption. The main function of the large intestine is absorption of water and electrolytes and formation of stool. The large intestine has several segments, from proximal to distal, cecum, ascending colon, transverse colon, left colon, sigmoid, and rectum. Much of the large intestine is intraperitoneal, so that it is covered by a serosa, but small areas are actually plastered against the posterior wall, such as the cecum and a portion sometimes of the sigmoid colon. So a portion of these segments are actually extraperitoneal and have an adventitia, not a serosa. In addition, when we get way down toward the anal canal, the intestine penetrates through the peritoneum. So the most distal part of it is also retroperitoneal or extraperitoneal and has an adventitia and not a serosa. Okay. Now, the other modification or break-in pattern that we see in the large intestine is a modification of the outer layer of the muscularis. So we've already mentioned that in the GI tract, our basic pattern is inner circumferential layer and outer longitudinal layer. But in the large intestine, the inner layer is circumferential, but the outer layer is modified into ribbons. So the longitudinal fibers are in these ribbons that are referred to as taenia coli. Taenia, a word that apparently means ribbon. So that's the modification on the outer layer of the muscularis in the large intestine. So let's look at the mucosa and see how this is specialized to achieve its function of water and electrolyte absorption. So you see this, and this is an image from Junqueira that shows this view of the entire thickness of the mucosa. So remember, mucosa is epithelium, lamina propria, which has these immune cells and loose connective tissue and blood vessels and nerves and lymphatics. And it has a muscularis mucosa at the deep edge, right? These tubular glands are lined by three major cell types: columnar absorptive cells, right? Colonocytes, goblet cells, and a few enteroendocrine cells, and actually a fourth type also, a few stem cells. Okay, so the two major cell types are columnar absorptive cells, also called colonocytes, and goblet cells. We've seen goblet cells before. They just produce mucin that has a protective and lubricating function on the surface of the mucosa. This is the real specialized cell type for the colon here, the columnar absorptive cell or colonocytes. And the modification here, you'll notice how this differs from the modifications we saw in the enterocyte, the small intestinal columnar absorptive cell. Remember, that cell was modified to do terminal digestion and absorption at the luminal edge of the cell, the apical side of the cell. So it had very well-developed microvilli on the apical membrane. Now, in contrast, the modifications for water absorption occur on the lateral side of the cell, where we get dilated intercellular spaces with folds of the cell with its epithelium extending into the dilated space. They call it interdigitating leafs of cytoplasm and membrane into this dilated space. And this provides the increased membrane surface area to allow the cell to maximize its function of resorbing water and electrolytes. You'll notice that the modifications on the apical surface are much less developed than what we see in the small intestine. These we would call blunt microvilli, right? Instead of the real well-developed microvilli that we see on the surface of the small intestine. Anal canal, the last segment. So the anatomy of the anal canal is surprisingly complicated. So I want you to just get a couple of concepts here. One is that, like the GE junction, the gastroesophageal junction, the transition between the rectum and the anal canal is another area of transition in terms of the epithelium. So at the upper level, we have the transition from the squamous line esophagus to the columnar line stomach. At the lower end of the GI tract, we have the transition from the columnar line rectum to the squamous line anal canal. And this transition occurs, for the most part, it's a little more complex, but for the most part, it occurs near the pectinate or dentate line. This is a landmark that is used by gastroenterologists and surgeons. You see it has the zigzag pattern here. And below this line, the epithelium lining the anal canal is non-keratinized, stratified squamous epithelium. Above the line is a small, what we refer to as a transition zone. And then above that is a columnar line mucosa, similar to what we see in the rest of the large intestine. And the histology here is illustrating that junction. This is columnar line mucosa like we would see in the large intestine. And this is a stratified squamous epithelium that we would see beneath or below the pectinate or dentate line. Now, the other transition that occurs is more distally, right near the zone where we have anal skin. And this is referred to as the white line or Hilton's white line. And this is where this epithelium becomes keratinized. Some of it may be keratinized stratified squamous, and some of it is actually skin. But in most individuals, it's actually hard to see that distinction. So you can just say that at the very distal end, the lining becomes keratinized and merges with the perianal skin. Okay, but the key transition zone is actually this pectinate or dentate line, in which beneath that line is non-keratinized stratified squamous epithelium, and above it is a little bit of what we refer to as transition and then columnar lined mucosa, like we see in the remainder of the large intestine. Okay? Okay, so that's the GI tract. So do we have any questions on that stuff? Okay, so the next segment are the accessory glands. All right, we're going to start with the salivary glands. Oh, wait a minute, Alicia has a question. Alicia, what
Hi,
is your...
Dr. Bennett.
Hi there.
Hi. So when you were talking about the columnar cells being absorptive versus
Yeah,
making mucus,
yeah,
when we say enterocytes, does
yeah.
that only apply to the absorptive cells, or would that apply to both?
Yeah, I really mangled that. So let me try to let me take another stab here. I want you to notice the word absorptive, right? And and I want you to contrast that with the stomach. You know, when you look at the histology just quickly, they kind of look similar, but they're really different. So in the small intestinal cell, you've got the brush border, right, which is what we can see on H&E that represents the location of the microvilli and the glycocalyx that covers it, right? That is present in the small intestinal cells, which we call enterocytes. The word enterocyte just means that it is a cell of the small intestine. Usually the entero refers to the small intestine, cyte is just cell, right? So it does happen that that word means it's a cell line of the small intestine, but those cells are columnar absorptor cells, okay?
Okay, got it. Thank you.
But I also want to say that the columnar cells are in the colon are also absorptive, but they have a different function. In the small intestine, they're absorbing and digesting nutrients. In the colon, they're absorbing water. And so morphologically, the modifications are different. Okay? All right. Good. Any other questions? Okay, so we're going to go on to the accessory glands. The first group are the salivary glands. So the functions of the salivary glands are moistening and lubricating ingested food and the oral mucosa. Initial digestion of carbohydrates through the production of salivary amylase, and secreting innate immune components like lysozyme and lactoferrin. Lysozyme, you can see, is an ongoing theme in the GI tract because it's antimicrobial and it's produced at multiple levels of the GI tract. Lactoferrin is interesting because it binds iron. Iron is required by lots of microbial organisms, and so by depriving them of iron, we make it less difficult for them to survive in the oral cavity and the GI tract. The major salivary glands that we're going to be focusing on here are the parotid, which is predominantly serous, the submandibular, which is a mixed gland containing both serous and mucus components, and the sublingual gland, which is also mixed but usually is predominantly mucus. The minor salivary glands are scattered throughout the oral cavity, and they are unencapsulated glands, which are usually mucus. Okay, so those are our major glands that we're going to be looking at. There's one little wrinkle, which if you've carefully read Joe Carey, you might have caught, and that is this issue of the initial site of digestion. Carbohydrates do begin digestion in the oral cavity. Lipids, there's some question about. The salivary tissue does produce, or the oral cavity tissue does produce a lingual lipase, which can digest fat, but most of the initial digestion of the fats does occur in the stomach. John Kara says both things. If you look at the section on the oral cavity, it says the initial digestion of lipids occur in the oral cavity. But if you look at the section on stomach, they have a comment that says the initial digestion of lipids occurs in the stomach. So when there's this kind of ambiguity, you can be sure that we're not going to give you a multiple choice question in which we force you to choose between them. Okay. Next. Does that make sense? Okay, organization of the salivary glands. So first we're going to talk about the secretory unit. Salivary glands are exocrine glands, so they produce a secretion in the secretory part of the gland, and then they transfer that secretion from the secretory unit of the gland to its destination through the conducting unit or the duct system of the gland. So first, with the salivary glands, we're going to look at the secretory component, and first we're going to look at the cell types that are present in the secretory unit of the salivary glands. There are two basic cells that produce secretion. The serous cells, which are pyramidal-shaped protein-secreting cells with round nuclei, basal rough endoplasmic reticulum, and apical secretory granules. The secretory granules of these serous cells generally contain amylase, lysozyme, a proline-rich antimicrobial protein, and the secretory component for IgA. The mucus cells, in contrast, are more columnar in shape, and their nucleus is pushed to the outside of the gland and is usually flattened rather than round. The cytoplasm is packed with secretory granules that contain hydrophilic mucin. On H&E stained sections, these two cell types usually look different. The serous cells, the cytoplasm looks more dense, and usually, depending on the preparation, will stain a purpley or bluey color. In contrast, the mucus cells, the cytoplasm often is almost clear staining, or sometimes stains a very light blue color. So they have a different appearance on the hematoxin and eosin stain. The third cell type that we see is the myoepithelial cell, which is present at the periphery, often in secretory, containing glands, and in the first segment of the duct system as well. So let's look at the ultrastructure of a mixed gland. So this is a gland that has both mucus cells and serous cells. The top of the image has several mucus cells, and the mucus cells contain large cytoplasmic granules which contain mucin. So the electron density of the mucin is intermediate compared to the high electron density of the serous cells, which contain the protein product of the serous gland. So these are mucus cells with large cytoplasmic mucus granules. Now the bottom half of this gland has serous cells. And the serous cells have smaller granules on the apical half of the cell that are more electron dense, that contain the protein products of the cell. But the lower half of the cell, the basal half, doesn't have secretory granules. It has rough endoplasmic reticulum, because these cells are protein factories, right? So they've got lots of RER on the basal half, and they've got secretory granules on the apical half. At the very base of the gland is a spindle cell right here, and that is a myoepithelial cell. Okay? Okay. Next one. How do we organize the cells in their secretory gland? So what is the histologic pattern of the secretory unit? In general, the serous cells generally organize themselves in an acinar pattern, meaning they appear on the hematoxin and eosin stain section as circles of cells, which corresponds to a three-dimensional structure that is spherical. And we refer to this as an acinar pattern, and the sphere as the acinus. On the other end of the spectrum, we have the mucous salivary glands, which often organize themselves into tubules or cylinders. And so this is often present as a mixture of both tubules and acini. So we call that pattern a tubulo-acinar pattern. Now, in the middle is the mixed gland, which often has a combination of two different cell types, serous and mucus. And it also has a mixed histologic pattern that is tubulo-acinar. So this drawing here from Junqueira illustrates this mixture of round acini and cylindrical tubules. Okay, let's look at another image here of this. This is blown up from Junkera, and we've highlighted the secretory component of the gland in yellow on the outside. And this secretory component is mixed. It's mixed both in terms of cell type with these bluish staining serous cells and yellow staining mucus cells. And you'll notice it's not just the color that's different. The serous cells have round nuclei sort of in the center of the cell, but the mucus cells have flattened nuclei that are at the very basal end of the cell. Okay. The other component that's illustrated here is the beginning of the conducting portion of the gland, or the duct system of the gland, and that's highlighted here in light blue. And the initial segment of the duct in the salivary gland is the intercalated duct. And then after that, it feeds into the next segment, which is a very important segment called the striated duct. So let's take a look at the different histologic appearance of these three types of glands. The serous gland, which is the parotid gland, predominantly serous. The submandibular gland, which is a mixed seromucous gland. And the sublingual gland, which is usually predominantly mucus. So if we show you a histologic section, a parotid gland section would look like this on the left, where every acinus seems to be circular in cross-section and has serous cells with round nuclei and kind of a blue to purpley cytoplasm. In the middle, we have the situation in the submandibular gland, which typically will show a very mixed pattern of bluish, purpley serous cells and much paler staining mucus cells. So we have a mixed seromucus pattern, and histologically we have a mixture of acini, like this round circular pattern here, and tubular acini, like this more cylindrical pattern here with a round bulb at the end of it. So that would be tubular acini and acinar histologic patterns. Now, interestingly, we also have an example here of a very important element of the duct system. This is a duct. This is an example of the striated duct. We're going to talk more about that in a minute. And the last example is this one on the right side. You see most of these acini and tubules have very pale stained cytoplasm and very peripherally located nuclei. So these are mucus glands. And the example of this most commonly seen in the sublingual gland. Okay. So let's talk about that conducting unit, the duct system. The ducts can be both intralobular, where they are right next to the secretory component, or they can be present in connective tissue septa when we call them interlobular ducts. So the intralobular ducts are the intercalated duct, which is lined by low cuboidal epithelium with a prominent component of myoepithelium at the periphery, and the very important striated duct segment, which is lined by more columnar epithelium that is specialized for modifying the primary saliva of the salivary gland. In the interlobular ducts, these are larger ducts that are excretory ducts that are lined by a variable component of epithelium. You can almost have any type of epithelium lining these ducts, simple cuboidal or columnar, and then stratified linings that can be cuboidal or columnar or pseudostratified. The main excretory ducts, the largest duct in the duct system, can become actually stratified squamous when they reach very, very close to the terminus of the duct. So when it gets very close to that oral mucosa, it can actually be lined by non-keratinized stratified squamous epithelium. So let's look at more closely at the really important segment, the striated duct. The striated duct is modified to modify the tonicity of the saliva and also the electrolyte content. So the striated ducts resorb sodium and chloride from the initial secretion, and they add potassium and bicarb. So the final saliva is actually slightly hypotonic and also is a little more alkaline than the initial secretion. Morphologically, the striated duct is characterized by infoldings of their basolateral membrane that are aligned with numerous mitochondria. So at the ultrastructural level, as we see here, you can actually see these. This is the basal side of the cell. This is the apical side of the cell. You can see there's a lumen here labeled L. So we know this is the apical side of the cell, and this is the basal. And you can see the basal membrane. You see all these folds in the basal membrane. And this cartoon shows that the folds actually are associated with numerous mitochondria. So the folds are modifications in the membrane that allow the cell to perform their function of modifying the electrolyte content of the saliva. And to do that, they have to pump ions across the membrane, so they need energy. So the basal infoldings are associated with mitochondria to provide energy for this transfer of ions across the membrane. Now, the ultrastructural modifications are reflected in the H&E light microscopic appearance of the duct. So these two images here are showing you what the H&E appearance of the duct is. And both of them are characterized by round nuclei that are in the center of the cell, halfway between the apical edge and the basal edge of the cell. And they're also characterized by basal cytoplasm that has a striated appearance. You can see it nicely here in this tissue section, and you can also see it even more dramatically in this one. So this is an example of where at the light microscopic level, what we're seeing is the evidence of the ultrastructural feature of basal membrane infolding associated with numerous mitochondria. That's what we're seeing in the basal half of the cell on H&E stained sections. Okay. Any questions about the salivary glands? Okay. So let's move on to the pancreas. Okay. So the pancreas is an example of a mixed gland. It is a mixed exocrine and endocrine gland. All right. So let's see. All right. So this image shows both a gross diagram of the pancreas nestling within the C-shaped sweep of the duodenum, and also a very low power histologic section, which allows you to see both the exocrine component and the endocrine component. The exocrine component is labeled A for acinar. This is the pancreatic acinar tissue. And the endocrine component is labeled I for islets of Langerhans. That is the endocrine component of the pancreas. Okay, let's see. So let's look at this diagram of how the endocrine component of the pancreas works together with the exocrine component of the pancreas. You can see that the endocrine component is a very small proportion of the total volume of the pancreas. It is in the structures called the islets of Langerhans. The islets of Langerhans are populated by cells that are smaller than the exocrine pancreatic cells, and there are various different cell types, some of which are listed: alpha cells, beta cells, delta cells, and PP cells, each with their own distinct protein or hormonal product. Beta cells, of course, produce insulin. Alpha cells produce glucagon. D cells or delta cells produce somatostatin, I think. Exocrine cells, the exocrine gland, is composed of the acinar glandular cells, central acinar cells, and you'll see that the intercalated ducts in the pancreas actually extend into the acids. So the acinar cells here in this drawing are yellow. They're here, right? The central acinar cell is light purple, as is the intercalated duct lining cells here. And this is the islet of Langerhans with the light blue parenchymal cells, and capillaries, because this is an endocrine tissue, so the parenchymal cells are closely opposed to capillaries. Okay. Let's look at some of the features of the exocrine pancreas, which produces 1.5 liters of alkaline secretions and delivers it to the duodenum. The acinar cells secrete proteases as inactive enzymes. They also secrete a pancreatic amylase, lipase, and various nucleases. The central acinar cells are actually the initial cells of the pancreatic duct system. So they're the initial cells of the intercalated duct that are actually present within the lumen of the acinus. So this is a distinction in the structure of the pancreatic acinar gland, the exocrine pancreas, and the exocrine salivary glands. In the salivary glands, the duct system, we don't see it actually in the lumen of the secretory unit, the acinus. Whereas in the pancreas, we actually see duct system cells inside the acinus, and those are called the central acinar cells. The intercalated duct in the pancreas is a simple lining, either a simple squamous or low cuboidal epithelium, and this is an important structure because it's responsible for secreting fluid rich in bicarb. So this is the component of the pancreatic secretion responsible for altering the pH of the intestinal contents. More on the pancreas. Pancreas has lots of mechanisms that protect it from autodigestion. One of these is the fact that proteases are synthesized and secreted as inactive enzymes, and they're only activated when they reach the duodenum. One of the mechanisms of activation makes use of the fact that the pK of these enzymes is higher than the gastric chyme's pK of pH 1 to 2. It's actually more like a pH 6. So we need the secretions of both the Brunner's glands in the duodenum and the pancreatic ducts to raise the pH from very acidic to more neutral so that these enzymes can be active. The other mechanism we use is a cascade of activation that utilizes trypsin. So trypsin is synthesized and secreted with a trypsin inhibitor. So that prevents trypsin from being activated within the cell. But once the trypsin is released in the duodenum, it encounters an enteropeptidase, an enzyme that acts on proteins that is located within the small intestine, and that activates trypsin. Activated trypsin is then able to activate the other digestive enzymes produced by the pancreas. So all these mechanisms prevent autodigestion of the pancreas until the digestive enzymes reach the lumen of the small intestine. How is this exocrine secretion regulated? Hormonally and autonomically. The hormones involved are cholecystokinin, which stimulates enzyme synthesis and secretion by the acinar cells. Cholecystokinin is produced by I cells in the small intestine. And the other hormone is secretin, produced by S cells, which stimulates water and bicarb secretion from the pancreatic duct cells. The other mechanism of activation is autonomic parasympathetic stimulation of secretion from both the acinar and the pancreatic duct cells. Now, one thing about the pancreatic exocrine gland, it is a serous gland, so it histologically may resemble the serous salivary gland, particularly the parotid gland, which is almost entirely serous. How do we distinguish them histologically? Histologically, we look for characteristic features. We note that the pancreas has central acinar cells and islets of Langerhans, and it lacks two features that we see in the parotid gland, myoepithelial cells and striated ducts. In contrast, the parotid gland has myoepithelial cells and striated ducts and lacks central acinar cells and islets of Langerhans. So if you see a histologic section of a serous gland, you can use those differentiating features to determine whether you're looking at exocrine pancreas or at parotid gland. And here's a little illustration of how we get the central acinar cell in the middle of the pancreatic acids. So if you look at the drawing on the right, this is a drawing that shows the relationship of the pancreatic acids. The acinar cells are labeled light blue in this drawing, with the yellow labeled duct cells of the pancreas. So this would be the intercalated duct. The intercalated duct extends directly into the pancreatic acids. So if we take a section through the acids, we're going to catch some of these intercalated duct cells in the middle of the acids. And when we do that, those cells are referred to as central acinar cells. So it's a word that describes exactly what they are. They're cells that are located in the center of the acids. So here's one here. Here's one here. This might be one here at the edge. And this is a nice H&E section that shows them as a nice differential appearance between the paler staining central acinar cell in the center of an acinus and the acinar cells themselves, which in this preparation are staining both red and blue. The zymogen granules in the apical part of the acinar cell are stained nicely red, and the basal part of the cell, rich in rough endoplasmic reticulum, is taking up the hematoxylin and is staining blue. Okay? So that's how we get the central acinar cell in the middle, the center of the pancreatic acids. Okay, any question about the pancreas? Next gland, the liver. The liver is the largest internal organ in adults, 1.5 kilograms or 2% of body weight. The liver is interesting also because it, like the lungs, it has a dual blood supply, right? So it has one blood supply from the portal system and one from the systemic system. The hepatocyte is the main parenchymal cell in the liver, and it's a really complicated cell because it has lots of functions. So let's just quickly go over some of these functions. It is involved in the production and exocrine secretion of bile. Bile is an exocrine product because it is secreted by the cell into a duct system, and that's the definition of exocrine. But the liver also produces products that are synthesized in the hepatocyte, but then are secreted in an endocrine fashion into the bloodstream. So examples of these endocrine products are many of the plasma proteins, albumin, fibrinogen, all of those apolipoproteins, and transferrin. Albumin quantitatively is the major product of the hepatocyte. The hepatocyte also engages in numerous biochemical reactions, such as gluconeogenesis, the conversion of amino acids to glucose, deamination of amino acids, by which we form urea, the waste product, and then it is very involved in detoxifying and conjugating toxins and drugs, respectively. These functions occur mostly in smooth endoplasmic reticulum. And finally, the hepatocyte has a storage function. It stores glycogen in glycogen granules. It stores fat or triglycerides in lipid droplets. It stores iron as the storage form transferrin, and it stores vitamin A in another cell type, the hepatic stellate cell. Another function of the liver, but not the hepatocyte, is the removal of effete, or old and tired erythrocytes, via the action of the liver's native macrophage, the Kupffer cell. So let's just take a look at this drawing for a minute, and you can see that the heterogeneous group of organelles reflects this heterogeneous group of functions of the hepatocyte. So if you just look at some of the things that we have in this drawing, we have lots of RER, rough endoplasmic reticulum, to synthesize proteins. We have lots of smooth endoplasmic reticulum to detoxify toxins and conjugate drugs. We have lots of mitochondria because all this stuff takes lots of energy. We have Golgi because we have to package our protein products. We have our storage forms of glycogen here and triglycerides, lipid droplets, right? And we also have the membranes of the hepatocyte, which actually, if you look at them, seem to have different appearances. We're going to get to that, I think, on the next slide. Oh, first of all, let's actually see if we can go back for a minute. Okay, so one thing about the liver, we've mentioned that we have both exocrine function and we have endocrine function. So the liver is kind of it often in histologic section looks like a square or a rectangle. And so some of these membrane surfaces are exocrine surfaces through which we secrete bile into a duct system. And some of the surfaces are endocrine surfaces through which we secrete protein products in an endocrine fashion into the bloodstream. So in this drawing, this surface here would be an exocrine surface because it has the beginning of the bile duct system, the bile canaliculus. And these two surfaces here and here would be endocrine surfaces of the cell because they're associated with the blood vascular space. On the bottom, the endothelial cells are labeled, and the vascular space on the other side of the endothelium is the hepatic sinusoid. This is a picture, I think it's from Junqueira, which is an example of rat liver. So now we're going to start talking about how we organize the hepatocytes in the liver tissue, the architecture of the liver. And this is kind of complicated, and there are a couple of schemes that we use to describe the organization of the liver. And this is illustrating one of them. This is from a pig liver. And the pigs have this unusual property in that when you stain them for connective tissue, it stains not only the portal triads, which of course are stained with connective tissue in everybody, and also the little vascular structure in the middle here, but it also stains connections between them. So it highlights the shape of this architectural structure of the liver. So this shape is a hexagon. And this hexagon is the architectural shape we refer to as the classic hepatic lobule. The classic hepatic lobule is a hexagonal lobule in which the vertices of this figure are occupied by a structure we call the portal triad. And the center of the lobule is occupied by a vascular structure we call either the central vein or the terminal hepatic venule. And in between them are radiating cords of hepatocytes, 1 or 2 cells thick. This is one of the structures we just alluded to. This is the portal triad. Thank you, Dr. Malvar. And this has 3 structures: the portal venule, the hepatic arteriole, and the bile ductule. One thing that's always interesting in terms of identification is the bile ductule often is of similar diameter to the hepatic arteriole, but the portal venule is invariably larger in diameter than the other 2 structures, and that sometimes can be helpful in identifying structures within the portal triad. This illustration shows the portal triad on the left, the central vein or terminal hepatic venule on the right, and the radiating cords of hepatocytes between them. The other structure that is visualized here nicely is that there is a space between the cords of hepatocytes that, as you can see, contains red blood cells. And this space is the hepatic sinusoid. So the hepatic sinusoid links the portal triad with the central vein. And when we want to see the structure of the hepatic cell cords in the liver, we will often do a stain that highlights the reticulin fibers on each side of the hepatic cords. And this stain is a reticulin stain. So let's take a look at this system of organization. This is from Junqueira. So you can see there are 3 systems of organization of the architecture of the liver. And what we've been talking about so far is the one on the left. This is a very important one. This is the classic hepatic lobule. And this is a scheme in which we organize the liver into hexagonal-shaped lobules, in which the center of the lobule is occupied by the central vein or terminal hepatic venule, and each corner of the hexagon is occupied by a portal tract that contains hepatic arteriole, bile ductal, and portal venule. Okay? The direction of flow of blood in this system is from the periphery, where the portal area is located, to the center, where the central vein or terminal hepatic venule is located. Okay? The second system of organization is one that is the least relevant for our study. It's called the portal lobule. I just want you to remember that there is a scheme of organization called the portal lobule. It generally is triangular in shape, and the center of this scheme is the portal tract. Okay? The third one is functionally probably the most important scheme, and this is referred to as the hepatic acinus. And the reason this is the most important clinically is that this, in a sense, is a functional method of organization, in which you can say by interpreting liver using this scheme, you get a sense of the function of the liver cells involved, and potentially, if there is hepatic cell injury, it can help you to determine the cause of that injury. So this scheme is called the hepatic acinus. And this scheme is actually, if you look at the shape, it is a diamond shape architecture in which the diamond crosses two adjacent classic hepatic lobules. The corners of the diamond are occupied, two of them, opposite corners, are occupied by a central vein or terminal hepatic venule, and the other two corners are occupied by portal triads. This is another drawing of two adjacent classical hepatic lobules with the hepatic acinus drawn over those two. You can see this is a diamond-shaped structure. Two of the corners opposite each other are occupied by central veins, and two corners are occupied by portal triads. Now, this is where the organization comes in. Using this scheme, we labeled the different zones of the hepatocytes based on their proximity to the portal tracts. So proximity to the portal tracts is also proximity to oxygenated blood in the hepatic arteriole and nutrient-rich blood in the portal venule. So the hepatocytes in hepatic acinar zone 1 have the most oxygen and the most nutrients. The hepatocytes farthest away from the portal area in hepatic acinar zone 3 have the least oxygen and the least nutrients. Now, functionally, this is very important, because if your patient suffers an insult where they're deprived of oxygenated blood, say they have an acute cardiac event and they go into an acute congestive heart failure, the zone of the hepatocytes that is most vulnerable is the zone that is furthest from the source of oxygenated blood, which would be zone 3. And that's why these patients tend to develop hepatocellular necrosis in hepatic acinar zone 3. Okay? On the other hand, if the patient has, say, a blood-borne infection, the zone of the hepatocytes that will be initially exposed to microbacterial agents will be zone 1, because these hepatocytes are closest to the systemic blood supply. So you can infer, sometimes you can infer pathologic causes of particular hepatocellular injury based on its distribution across the zones of the hepatic acinus. In the liver, blood flows from the portal areas to the central vein. But in contrast, bile flows in the opposite direction, from the parenchyma of the portal lobule to the portal tract. So blood and bile flow in opposite directions. Let's just take a look at the biliary drainage system of the liver. So just like the duct system we were looking at in the exocrine glands, in the biliary drainage system, we have the initial segment of the duct system is the smallest in diameter, and in the biliary system, it's also the simplest. As we progress toward the portal area, we get a larger diameter duct until we arrive at the bile ductule in the portal tract. So let's first take a look at the smallest element of this duct system, the bile canaliculus. So this is most easily seen at the ultrastructural level, which is this image on the left side here. This is a transmission electron micrograph that is illustrating a bile canaliculus labeled with the star. And the thing about the bile canaliculus is it does not have a separate epithelial lining. The edge of the canaliculus is formed by the cell membrane of two adjacent hepatocytes. So this is one hepatocyte, and this is a second hepatocyte. And each of the hepatocytes on their membrane has a groove which matches up with a groove in their neighbor's membrane. And then we have tight junctions that seal off the edge of this groove on both sides. And this forms a tube, which is the bile canaliculus. So the bile canaliculus has microvilli from the hepatocytes, but no separate component of lining epithelial cells. Okay? As the duct gets larger, the next segment is referred to, so this is the canaliculus here without lining epithelium. When it acquires lining epithelium, we refer to that segment as a bile canal of Herring, which is right here. And as they get larger and larger, they spill their bile into a bile ductule, which is located in the portal tract. And of course, these bile ductules connect in arborizing fashion into larger and larger bile ducts, eventually ending up at the cystic duct to the gallbladder, or the bile duct which extends through the ampulla into the duodenum. So let's just say a few words about the gallbladder and biliary tract. The gallbladder is a sac-like structure that, and here's the key, its function is to store and concentrate bile. Okay? It releases it via the biliary duct system into the duodenum after a meal. That release is stimulated by a hormonal mechanism through cholecystokinin that we've already talked about. So just remember, function of the gallbladder stores and concentrates bile. So as a result, since it has a concentrating function, the cholangiocyte is going to be modified in a way that will allow it to concentrate bile. So this cell, in a sense, is analogous to the colonocyte that we talked about. If you remember, one of the major functions of the large intestine is to remove water from the luminal content and form stool. And so the colonocyte is modified with lateral intercellular spaces with folds of the cell into the space where the transfer of water and electrolytes occurs. So in a similar fashion, the cholangiocytes lining the gallbladder also are specialized for water uptake. They remove water from the lumen into the interstitial space of the tissue in the gallbladder, and they do this by folded basolateral membranes in a dilated intercellular space. So it's similar to what we saw in the lining of the colon. Now, the histology of the gallbladder is interesting because this is another break-in pattern from the GI tract. It's a break-in pattern because although it has these kind of finger-like folds in profile, these are not as regular as intestinal villi, and they are referred to as a folded mucosa. So it's a different pattern of specialization of the mucosa. This is a folded mucosa, not a mucosa lined by intestinal villi. Also, it has a lamina propria is present, but there's no muscularis mucosa, and we don't usually have a distinct submucosa in the gallbladder wall. Instead, the mucosa is directly adherent to the muscularis, and the muscularis is unique or different because instead of having two distinct inner and outer layers, it actually has muscular fibers that extend in multiple directions. So it's a different architectural pattern of the gallbladder wall than we see in other areas of the GI tract. That's it. Anybody have any questions about any aspect of this? It's question time. Anybody? No?
Thank you so much, Dr. Van. Would we have access to the slides?
Yeah, I think we can post them. I can
You guys
post
have
them.
all of these slides already, but okay. The
We
same
do. There's
stuff you have.
slight modifications on them. I mean, they are actually slides that we've all seen. But I can post them. I can send them to Dr. Malvar and to Dr. Luis. But I can post them on Canvas at the Middletown campus. I don't have access to the other canvases. Is that the plural of Canvas, Dr. Malvar? Canvases.
Canvases is fine.
Not Canvas I? Not Canva?
I don't know.
Okay, all right. Anybody else? No. You guys, oh, wait a minute. Oh, Canvi. Alan Bass says Canvi. Okay, all right. Anybody else have any other
That's
suggestions?
not good.
Oh, he's joking. Okay, yep, gotcha. Anybody else? I guess we're done. Everybody, good luck on the exam. Remember, it's another week until we get there. So we have lymphoid tissue next week. And of course, we're always available for questions. Okay. All right. Thank you, everybody. And thank you,
Thanks.
Dr. Malvar. And we'll see you.